Start my presentation. Good morning, all. Thank you for joining. So now I'm going to walk you through the basics of genomics. Then what are the applications of genomics and the opportunities in genomics. So to start with, we're going to start with introduction and evolution of sequencing methodologies, application of NGS technology in genomics, and NGS workflow overview, genomics application, carrier in genomics. Here it will be like a skim through. Hello. Can I continue, guys?
Yes, ma'am.
Okay, thank you. So what is a genome? Genome is nothing but a haploid set of a chromosome in case of eukaryotes, but in case of bacteria, it is a single chromosome. When it coming to the virus, it is a DNA virus or a RNA virus. Genome is denoted by X. X means indicates your ploidy level. Genome depends on your ploidy level. If you can see here in the example, given in Drosophila, see here, this X is X to X. X indicates your ploidy level. N indicates the haploid level. So genome X of a Drosophila is 4. So the term genome was introduced by Winkler in 1920 and denoted the complete set of chromosomal extra chromosome gene present in an organism. So what is genomics? Genomics is nothing but a study of genetic material. In the genomics, we study entire coding and non-coding genetic material. But when it comes to the genetics, in genetics, we study only the genes. That is the coding part of our genetic material. So genomics, as I told you, it takes into account both coding and non-coding regions. And what are the main things involved in genomics is sequencing in the current situation, sequencing, analysis, and interpretation. As you can see here, see this is my cell. This is my whole one cell nucleus. Just it has been expanded into prophase of your cell division where your condensation of chromosome happens. And my DNA is bound into the histone proteins. And when you enlarge that, you can see your double-stranded DNA where your nucleotides are bound together. Because this what in the genomics we study is what is the sequence order and what is its function? What if alteration happens in the sequence? Those things are addressed in genomics. And to study genomics, sequence is a technology to understand the pattern of my nucleotides. So as I told you, sequence is next generation sequencing or sequence is technology to address my genome. How this sequencing technology evolved? As you all know, the first generation sequence that is in Sanger sequencing. In the Sanger sequencing, that's the first generation sequencing where we usually clone individual small fragments, then sequence. Bioinformatically, we will stitch, then we do the analysis. And it was taking a long time. You think, guys, cloning, again, seeing that whether my clone is there, then sequencing that, stitching it is too tedious also. And then maybe its error also could be incorporated. Then second generation sequencing is your Roche 454, which is no more in the market. So Illumina, Ion Torrent in AB Solid is also not. That is my second, short reads. Basically, my second generation sequencing is my shorter reads. And third generation sequencing, we have PacBio, Nanopore, where it is also called as long read sequencing. So how is the evolution based on the year, my sequencing methodology? My Sanger came around between 1975 to 1970. Then till almost till 2000, 1995, between 1995 2000, we got our, 2005, we got our second generation. And after 2005, we got our introduced third edition sequencing technology. So what is the applications in NGS technology? So in application, when you come, the starting material will be either DNA or RNA in sequencing. When I take the starting material as DNA, so what are the things I'm addressing? The genome, what we talked is a haploid, what we sequence analyze a haploid set of your genetic material. Then this epigenome, which is away from Mendelian genetics. What happens in epigenome, the structure of your DNA, there is no difference in the sequence of your DNA. Only there is chemical modifications of your DNA, which is also important for the expression of your genes. So when I come to the genome, in the genome, I can target my small fragments called as target sequence. Then either I can do whole exome sequencing, where that is my coding region, and whole genome sequencing in WGS, where entire genome is sequenced, and metagenome. Metagenome we used where there is an environmental sample or a combination of microbes are present. So in target sequencing, it is kind of resequencing, because when I have the target sequence already, I know my target. I know my target sequence, so it is called as resequencing. And further analysis go with mapping. And what is the objective of my target sequencing is I want to identify my SNPs or the variations, genetic variations. In whole genome sequencing, it is like a de novo sequencing. So, or it can be a reference also. If it is anyway de novo sequencing, because the reads will be there, we have to assemble the reads. And once we get the assembly, I have to know what is the each portion is doing. That is annotation. What protein is coding. That is called annotation. Then any other unannotated genes are there. If it is information is not available, that is called a putative annotations. Then epigenome also. The histone modification and the two genetic modifications, histone modification and DNA methylation, the basic principle in that. In histone modification, we go for reference, this one, resequencing and de novo assembly, and analysis almost remains same. Assembly or mapping is same. Then if my starting material is RNA, that is called transcriptome. In the transcriptome, we do whole transcriptome sequencing, long non-coding RNA sequencing, short non-coding RNA sequencing, target sequencing. As you know, in RNA, we have both coding and non-coding region. In whole transcriptome sequencing, if it is a de novo, again assembly goes. If it is a good reference is available, good. If the good reference is available, it will be like reference-based alignment. Then there in the de novo, we see identify the transcripts, and it goes further with other downstream analysis. In target sequencing, I do the mapping and do the variation, identify the variations. So if you come to the overall NGS workflow, this is like a skim through. First is the sample extraction. Before sample extraction, sample collection is also very, very important. Because if my sampling is good, I can get a good biological molecules like RNA, DNA, even if you go for protein also, If my sample is not good, if my RNA, DNA starting material quality is not good, it affects my library preparation. So the collection of sample is sample, and the transportation of the sample also matters. Storage, transportation, everything matters. So when you come from if you join the training program, we will teach you from the sample collection, then how to store it, how to process it, how to send for the labs. So next steps come the extraction, and even the extraction method is not universal for all the samples. So each sample type there will be some composition variation. So in the training programs, we will teach you how to handle different samples and how to which method to be used. Then quality control in every step it will be there. In library preparation, the next comes the library preparation where you extract your DNA. Your DNA will have blunt and or sticky ends. So basically we do the end repair, because if you don't do the end repair, the adapter doesn't ligate. So adapters are nothing but the known sequence where it helps in the sequencing. So when you come for a training program also, all these steps will be dealt in details. After my library preparation and quality control passes, then I go for sequencing. Sequencing also, here also we teach you which objective, which application, what platform to use, and whether I want a longer read or a shorter read. So all this will be dealt when you come for training. Then analysis, based on whatever the people want, they want a de novo, or the trainees want de novo or transcriptome or reference based or target based, which is an amplicon-seq. We'll teach you the analysis pass. Also, it depends on the duration people join for the training program. So what are the applications? In which field this genomics is applied? So you can go, scientists are using in address the evolutionary and conservation biology. Data analysis and computational tools also can be developed. And in the for Sonics lab, there also we do the DNA fingerprinting, food and beverages, there to see any contamination, or even when the product is in the product, for example, in the Ayurvedic stuff, they list out like 20 plants. The 20 plant extracts are involved in the whatever the medicine or whatever the thing you take, boosters you take. So through genomics, we can also validate it whether all this composition is present or what, or in the food any adulteration is there. And in clinical healthcare, now it is booming up. Personalized medicine or gut micro transformation, everything is happening. And in agriculture also, it is helping a lot. To be honest speaking, in agriculture, if I want to develop a variety, maybe like a fortified variety where to enhance my nutrition, or good yielding variety, in a traditional way, it takes 10 to 12 years for me to release a variety for the farmers to cultivate. But using genomics application, within 2 to 3 generations, maybe within 2 to 3 years, with a multi-location trial, I can come up with a good variety to meet the demands of the growing population. And in microbiology, microbiology is like everywhere. And to take from environmental microbiology to dissolve all our waste, or there's biofortified products available, and there are resistance genes which we can import in agriculture crops, and biofertilizer. What is you're talking about? Organic, everything. It's like this microbiology also is like growing up like anything where the genomics application is having its great impact. And mitochondrial genome in the eukaryotes, where it is maternally inherited. Coming that whatever I explained, like in the healthcare, gene discovery and diagnosis of rare monogenic diseases. Like monogenic diseases are those diseases which are controlled by single gene. What I mean to say is, in the training program, we not only use, for example, if I'm using monogenic disorder, you believe that is a technical word. What do you mean by monogenic? It is controlled by single gene. So not only learning the genomics, you also learn the genetics of each of the things, material which we teach. Identification and diagnostic of genetic factors contributing to the common diseases like diabetes, it may be now, like those things we use. Pharmacogenetics and target therapy. Nowadays, especially in the cancer genetics also, it is coming up. I identify the mutation, and now we are talking about the CRISPR also, where I can correct my sequence, and that can be like the personalized medicine. Again, it comes to personalized medicine only. Then I can cure my disorders or diseases. And prenatal diagnosis and testing. And in case of infectious disease, I think recently, like 5-6 years back, there was an E. coli outbreak in Europe. So using the nanopore technology, whoever was coming for the airport, they were able to do the sequencing on the spot and see whether there's an E. coli infection. So because that is the beauty of genomics and the sequencing platforms. And as I already mentioned about personalized medicine, gene therapy, genome editing, which is a CRISPR technology, and HLA typing, where there is an organ transplant to match between the patients and the donor. In agriculture, my genome size is also important because my plody level speaks a lot. And gene number, gene mapping, gene sequencing, gene cloning, DNA marker identification to identify those plants which have the favorable traits. If I want to see a particular trait in a plant, if I have a DNA marker, instead of doing whole genome sequencing or thing, I can just develop the SSR markers, SNP, screen my population. And as I was talking, marker data selection is the selection where I was telling combining the genomics and the traditional plant breeding. And transgenic bleedings, here we can see where my insertion is, what is going to do to my host genetic material and other things. And linkage map, QTL mapping. These QTLs are the genic markers. So it will be help for screening of the germplasms, because those are like genetic reservoirs for us. And application again comes with data analytics computation. As you know, in genomics, a lot of data will be generated to the next generation sequencing. So a lot of computational, and you had to analyze the data, process the data, interpret it, and many of the algorithm developments will be there. Sometimes it will be the custom design. All the algorithms or all the tools are not same for a thing, because each biological system is different. So there's a scope over there also. And evolutionary conservation biology, I told you the genetic diversity. If I have the diversity in any population, it is easy to address any diseases or any problems in the particular population. And the mitochondrial DNA, as we know that mitochondrial DNA is passed on from mother to the progeny. And nowadays, some of this important traits are also can be traced to mitochondrial genomics. And in case of forensic labs, to see identify the criminals, or it is also growing up. In microbiology, as I discussed already, with the environmental samples, human, animal, and for biofuels. And genetics, in the case of human microbe, when I do the human microbe genome sequencing, it promises to cure many diseases and identify the disorders also. In animal microbiome, it helps to improve the livestock health. And soil microbiome, as you know, like the soil health is depleting a lot, and which will have the crisis for our food production. So it promotes sustainable agriculture through my data analysis and to see what are the microbial composition in the soil, and suggest us or the scientists to see how the soil health can be improved and what the preventive measures can be taken to solve the further problems of food or whatever it is. And food and bread industries, I already briefed that, to see the adulteration or to see whether the composition is right, whatever is mentioned in the label. So why I should pursue the career in genomics? See, because it is in high demand. When I say high demand, every field, take from the fossils till the living organisms, genomics is applicable. When there's a high demand, there are a lot of opportunities even in the industries or in the clinics or in the academics. Everywhere when there's a demand, there's opportunities. And genomics is like an ocean when you think. Research and innovations, because there are a lot of innovations to be done. And through research and innovations, we come up with a main methodology, new techniques, new tools, where we can reduce the cost or turnaround time, and to address different biological organisms, because all organisms, biological genetic material is not same. And when we have high demand opportunities and there's a research and innovation, there will be versatility in the career also. As I told you, one can go for clinical genetics, one can come for plant genetics, marine genetics, then you have these fossils, forensics labs, these are the versatile career opportunities. Genetics people, genomics people jump in any way there is organisms or a biology is there. So as a career, when you talk about career, there are, as you told, there are many other opportunities in genomics. So we can be specialized in one of the things. Better, if I have the thing of being in a wet lab, I can specialize in wet lab, because genomics, as it includes both wet lab and dry lab, I can get specialized in wet lab, or I can specialize in data analytics, and many other, like legality will come. So many other applications, many other career opportunities are there. For example, if I say like computational genomics and data scientist. So when I want to be in bioinformatics, if I get a opportunity or a career, if I want to go as a computational genomics and data scientist, see where the mathematical statistics is also very important in the designing an experiment. So what I'll do with there, I'll develop the pipelines, I validate the pipelines, and then whatever data comes, I do the analysis and give a biological interpretation for that. And technical developments, like as I told you, many tools has to be developed, even the dry lab, in the wet lab, many protocols, even if anybody is smart enough, another sequencing technology can also be developed where now we go with the like multiplexing the sample, even a single sample in a less time, if I can come up with a technology where and bring the cost down. So there's a lot of scope for the technology development. And for genetic researchers, can be academic, it can be corporate sector, it can be in clinical sector, where we do the lot. We are not understanding anything about whatever the organisms are. There are so much things to decode and understand what is going on in the living organisms. And then ethical, legal, and special implications. Ethical means because DNA, RNA, as you know, if I use my DNA, I can clone, right? So there also genomics person who has a knowledge of genomics can be in that committee and have all the rules and regulations. If I want to send a sample out, what are the pros and cons, and if I want to, if I have the genetic data, if I share, all these kind of views can be given for the ethical, people, and genomics people can also fit in the ethical, legal, special implications research also. Then genomic medicine clinicians. For example, clinicians like nurse practitioners or pharmacists or physics, genomics people who are specialized in genomics can explain them the results. And based on that, even a doctor can say, for example, it's a gut microbe. If you sequence a gut microbe, we can see which is what are the population of your good microbes and the bad microbes. And even you can see what antibiotics and what is the dosage it has to be recommended. So when if you want to pursue genetic counselor, because sometimes there are some genetic disorders or some of the rare diseases where the genomic specialized person can intervene and explain the patients or the parents or the patients what is it, educate them about what is happening, what will happen, what are the preventive measures has to be taken. And bioinformatics specialization, as you guys know, bioinformaticians are the people who combine the computer science and the biology. So they analyze the larger data, such as raw data. Then then they maintain the databases, they identify the mutations and say where it is annotated. Based on that, many other experiments also can be thought, or any in case of disease or anything, they can think how to treat them. Scientists in cancer genetics, as you all know, that now cancer is raising like anything. There the mutations, we can sequence, as we know already, cancer panels, few cancer panels are there. We can also come up with a different cancer panels, see the variations in the patients, and tell them what therapy. Even nowadays, scientists also thinking, if I can identify the mutations and the frequency of mutations, the position, we can say how much radiations or how many chemotherapies are required. And the rare diseases like thalassemia, sickle cell anemia, as it is from hereditary, it is transferred from progeny to progeny. We can also, before transferring also, we can say whether it will be transferred, what is the chance of it is transferring to the next progeny. There also genomic specialized people play a lot of role. So again, in the scientist being like in a higher position, scientist, bioinformatics, computational biology, you can, as I told you, like if you have a good background, you can come up with the tools, it is relevant to the biology thing. Production manager. Here, even in the production manager, also you can work in the instrumentation things, not only in the biology or in the computational, the computer field. People with a genomics background also can work on the production as a production manager, the instrumentation. They can also help in designing the instruments for use of in the genomics, give their inputs, involve in the designing and the production of those, and validating those things. Quality control manager, which is very, very required for all the genomics products. As you know, genomics is nothing but a part of molecular biology. When you say molecular biology, so many molecular techniques are involved. And in every technique, there is a quality control. So we can work as a quality control. And field application scientist, here, like for example, in any, for example, Illumina or in the PacBio, when they go and install the instruments, you can help them set up the run and teach them how it works, technique, principle of those things. And also they can be in the sales and marketing. So people who have genomics background go and talk about the products or the consumables for the library preparation or the instruments. So there's a lot of weightage because you talk in the biology background. So sequencer design experiments and engineer. Here also, like, and as I told you, like same, it's like another designation in case of whatever I told you, like here only sales, marketing or field application scientist. Sequencer designing, I can teach the scientist or the anybody who is in how to design an experiment. From designing experiment till loading onto the machine, I can also be in that field if I am expert in genomics. Lab manager or lab head, where you manage all the project analysis, quality control, and troubleshooting, protocol, guidelines, that is the position what it demands in the lab manager. Academics and research, teaching is also one thing. Along with that, we can continue pursue with the research of genomics. The regulatory affairs specialist, this is like individual. Now the specialized like drug designing also come in biotech industries, where insulin's production, and to maintain the genetic purity of an organisms which is used in the biotechnology to address many of the things. For example, it can be biofertilizer, it can be a drug designing, or it can be like from the microorganisms we go with the enzyme production, everything for those. Even because when my organisms have maintained a genetic purity, then only I can use for further downstream analysis. So this was an article in the paper, and also on one of the website, Nova Advisor. You can see 2023, Indian market is 1.8 billion. It is expected to grow in 2032 7.8 billion. And also there was an article in Hindu, if you have guys seen in 2023 about Indian genomics, how it is growing, and how government is helping. Not only it is a government is also encouraging for the genomics to address many of the existing problems, which we are facing now. So there is a lot of scope in genomics, but only thing is one has to get specialized. I had to get specialized. I had to get the training, understand what is going on, what I have to be in. If we understand all those things, surely we will be in a good position. So in BioNavid, when I skim through all those things, we have different. In BioNavid, not only this genomics training program we do, we also give the services. If you see to your right side, we have whole genome sequencing, whole exome, whole genome finishing with high-seq genotyping by sequencing, single cell exome, exosomal DNA sequencing, epigenetic, transcriptome, micro. Basically, I can say BioNavid is a one-stop solution from where we guide for the scientist, for anybody, how to collect the sample. Even we help with the experimental design. If they tell their objective, what they want to achieve, and what material they have, we hold their hand from experimental design till the publication. It's like a one-stop solution for the scientists. And even in that, when the PhD students or when a project comes, we would train the people what are all the steps in genomics. So in the area of genomics, we have 500,000 plus projects completed, 250 plus research articles published, in which we are 100 plus co-author, and 20 plus diverse taxonomy. This indicates how experienced we are in genomics as we are working in and out with genomics. So in coming to the training programs, we have different training programs. We have like your 1 week training program, 6 months, 3 months, custom design training program. Also, we help the dissertation. We help them like do the genomics work and write the dissertation, complete their degree. And even like sometimes if there is a need, if there is an opening in a company, if any trainees come to our training program, if they are very good, if they are very good in genomics, we do take them we do offer them job to work with us. So this is all overall, it's an overall scheme of genomics. What in genomics, what are the main things in genomics, what happens inside the genomics, and what are the opportunities, and what is the future in genomics. So now I can open thing for the questions. Anybody have questions, particular questions, or anything to be repeated? I'm happy to repeated. Thank you. Thank you for joining. Any questions, please? Hello, you can unmute and ask the questions. Hello, anybody have questions?
Hello, ma'am.
Yep.
Ma'am, thank you for such a great webinar. It was really nice to know about everything, ma'am. Ma'am, I wanted to ask, like, for the training program, who shall we connect to,
Mhm.
ma'am? Like, if we want to do it.
See, we have. Oh, sorry, I forgot it was last. Can you see here the contact information? Even if you go in the website,
Yes, ma'am.
you have this address,
Okay, ma'am.
and you can tell like what training you are looking for.
Mm-hmm.
And it
Mm-hmm.
will be great. If we do in batches, but if from your institution, if 5 people or anybody comes together, immediately we can start.
Okay. Okay, ma'am. Thank you, ma'am.
You're welcome.
So, Rupa, can we also brief whosoever has joined regarding our training programs, like what all trainings, like wet lab, dry lab, right?
So here to say like we do both the wet lab and dry lab training. Wet lab training, as I told you, as I told you in the middle of my presentation also, we start from the sample collection. So we teach you from sample collection, then extraction, library preparation, then data analysis, and even the interpretation. In dry lab training, it depends like what transcript or different application it is, DNA or RNA sequencing. We will train you in data analysis, interpretation, and generation of the figures, pictorial representation, which can be published. So it depends, but the duration varies. If you want both wet lab and dry lab training, it will be like 3 months to 6 months. Or if you want to just know simple individual application, like transcriptome sequencing, library preparation, it will be like in 10 to 12 days training program. It depends on what is your requirement. And also we publish the batches. When the batch will start, group of 5 or 6, like that.
And I think it is good for the graduate students as well as undergrads as well. So for PhD students and all, if they are working into certain NGS based platform and or they are working with some genomics related this thing, they have some thesis. So they can, and when you're doing a PhD, I'm sure that you would be interested into a particular application. They can, of course, they have to defend at the end the thesis by their own, so they have to have that understanding of what application you have used, be it a dry lab or wet lab, and what is being done there. So that our training programs will help those PhD students as well who are interested into learning, if they have incorporated some genomics application in their thesis. And it is also good for the graduate students, basically who want to basically who are looking out for the career, and they don't know where should we go, where we should look for. And if they have like background of some biology with them, so bioinformatics is and genomics is now a career to choose for, because as Rupa has already mentioned that there is a lot of scope, lot of opportunities out there. And this skill set wherein you are having uh, biology background with the informatics computational background as well, the skill set is still a niche skill set which is missing. So if you have that skill set, or if you develop that skill set through our training program, that is going to definitely give you a boost in your career. And there is lot of opportunities out there wherein you can, as per your interest, you can pitch in.
Hello, any more clarifications? So also, like any other clarifications are not there, you can reach out. The number is here, and if you can contact us also, mail us also, and tell us what you are looking, what you want. We will see how can we accommodate, and as Dr. Rowley told also, like anybody like want to wet lab people want to try bioinformatics, or bioinformatics to know what is going on in the wet lab, which helps in the data analysis. So you can also let us know and write and reach out us calling or writing a mail. So we will end up now if there is no questions. Okay, guys, then bye. See you. Reach it out. Reach us out. Thank you. Thanks for attending and listening to me. Thank
Thank
you.
you.